And what this new research shows is that if you do gene editing in mouse cells by a totally standard methodology, what happens is all this DNA sneaks in from places that nobody really expected it to come from.
You're listening to the Corbett Report.
Welcome, friends. James Corbett here, CorbettReport.com. Today is the 23rd of October, 2019, here across the dateline in Japan. But today we're talking to a guest who is a previous Corbett Report guest that I hope you may be familiar with. That's Dr. Jonathan Latham of IndependentScienceNews.org. And if you are not familiar, you can go back in the Corbett Report interview archives back to interview 1096 from 2015, where we talked to Dr. Latham about GMO concerns and a article that he had written at that time, Growing Doubt, a Scientist's Experience of GMOs. Today we're going to be talking about a related but slightly different subject, gene editing. And we're going to talk about a new report that he's just put out at IndependentScienceNews.org, which will, of course, be linked up in the show notes. Dr. Jonathan Latham, thank you again for taking the time.
My pleasure, James.
Let's get into your new report. Specifically, we're talking about gene editing unintentionally adds bovine DNA, goat DNA, and bacterial DNA, mouse researchers find, which is a very interesting article, but one that, does get technical, as a lot of your articles do. So for the non-technical, non-specialist audience out there, why don't we just start with the basic question, what is gene editing?
So, well, gene editing is a little bit of a misnomer. What it really is targeting cuts in DNA to specific locations in the genome, and then basically have the cell repairs those cuts, and researchers select from cells that have repairs that they like the look of. So if you're a researcher, you'll take a whole, set of tissue cultures, or you'll take cells that are going to turn into a new organism, and you will basically select the one that you like the look of. And and you can do that, you can select through as many organisms as you want, basically. So it's not editing in the way that you would do it on a keyboard, but it's something similar-ish.
So the gene editing industry, such as it exists right now, is touting the safety of this technique and the fact that it will not introduce foreign DNA into the gene-edited subjects. But of course, as the title of this article, and as the article in depth demonstrates, this is not the case. Tell us about the latest research that refutes this claim.
Yeah, sure. So there's a big fight going on at the moment over the regulation of gene editing and whether it should be, subject to the same constraints as GMO organisms. And, that debate is at the moment being settled in different directions, in different jurisdictions. So the U.S. is going kind of in favor of no regulation, at least if the USDA gets its way. And countries like the EU or blocs like the EU are looking at regulating gene edits as GMOs. So there's a big fight, and the industry, the breeding industry, there are all kinds of, there's a whole battery of entrepreneurs out there wanting to produce, additives for food. There are people who want to produce gene-edited livestock. There are people who want to produce gene-edited crops. They all have a stake in this regulation. And what is, their big claim is that this editing is so precise that essentially you can't tell the difference between an edited organism and one bred through normal, say, plant breeding or livestock breeding and so forth. And some people seem to have bought into this argument and some people don't. And but the big claim of it is that, at least in principle, you can make something that is negligibly different from the parent organism. And what this new research shows is that if you do gene editing in mouse cells by a totally standard methodology, what happens is all this DNA sneaks in from places that nobody really expected it to come from. So, for example, you need to add some DNA to your cell culture in the form of the gene editing machinery or the gene editing template that you want to alter the cells to, and it turns out that this DNA has contaminating DNA. The which comes from E. coli, right? So basically, when that DNA comes from E. coli, it means that DNA from E. coli, a bacterium, is basically going to end up in the mouse cell. What this new research also showed is that when they were editing these mouse cells, what they found was goat DNA and also cow DNA. And the reason is because they're reagents derived from living organisms, from cows or goats, and sometimes from other species that are also present, because they're kind of standard reagents that all biologists use, because, for the most part, they're not trying to commercialize a product. So essentially what's happening is techniques that are being used in the lab to make, single cells or to alter things that will never leave the lab are being adopted by these entrepreneurs. There's this company, Recombinetics, which a lot of this fuss is about, which is basically producing these hornless cattle. And these hornless cattle, it's claiming, are identical to hornless cattle developed by other methods. And they're using these standard lab techniques. And they are almost certainly going to have E. coli DNA and DNA from different species ending up in their animals. And so this is totally contradicting to the propaganda, if you like, of the biotech industry that, so much is resting on as far as they're concerned.
That's right, and that relates to an article that you put up last month, August. FDA finds unexpected antibiotic resistance genes in gene-edited dean-horned cattle, which is again talking about this Recombinetics company in particular and their polled cattle. Yeah. Just to clarify, this isn't a problem specifically with Recombinetics, this particular biotech startup. You're saying that this is actually the standard procedures for gene editing as it's applied right now.
Yeah, it's kind of interesting because you have people quoted in the media. This story has been picked up by various outlets. And you get people quoted in the media, some of them saying, well, we never expected this antibody, the cattle. So these cattle specifically were going to go to Brazil and be used in breeding programs, and Brazil's now closed down that breeding program and so forth. So there have been implications of this already. But essentially, some people are saying, oh, this is no surprise because, you put antibiotic-resistant genes in there, they're going to, and sometimes will end up in the edited organisms. And other people are saying, this is a total surprise. We never imagined that this would happen. And Recombinetics basically did not look for the kind of changes that they found. Basically, it was the FDA that found the changes that Recombinetics had put in there. And Recombinetics is now on the record as saying, we didn't, I mean, to me it's insane, but they basically said, we didn't think to look for this.
That is insane. That would be the first thing that I think any layman would expect about this is that, Recombinetics found the problems, but you're saying they didn't even look for those problems.
No, it seems, I mean, there's two interpretations of why it took FDA 2 years to find it and Recombinetics never found it. These animals were born 3 years ago. This has only just come out in the last month or two. So, one interpretation is Recombinetics is simply incompetent. And, in terms of their, looking for DNA in their computer programs and their algorithms and stuff. And the other is that their board of advisors and their scientific people basically didn't even think to look. And it seems the answer was they didn't even think to look.
That's startling and shocking, really. And what are the implications of this finding? For what does it mean that bovine DNA, for example, is ending up in mice? What are the possible ramifications of something like that? Yeah.
Well, on one level, you can say, well, this is just mammal DNA getting into another mammal. Maybe that's less interesting than E. coli DNA getting into a mammal. But, organisms are basically organized in such a way that they basically are used to various genetic events happening in the process of reproduction, and incorporation of DNA from foreign organisms is not part of that recipe. So, you can think of just general random things that might happen, that genes become misexpressed, they have, health problems, maybe that would be upsetting, or people are making gene-edited crops that have then produce toxins or some molecules that are not good for your health, for example. So, there are potential toxicological consequences for people. And we have, there are examples in the world of GMOs of inadvertently introduced genes that were basically, or either known to be or thought to be toxic to the kind of organisms that were going to be fed these crops. So that's one possibility. But also there's recombinetics, the bovine DNA ending up in the cow genome basically at random. That was never, basically random DNA or semi-random DNA ending up in these animals means that you have the possibility of transferring viruses, for example. They found transposon. They found basically, viral DNA from retroviruses was ending up in the wrong place of the genome of these mice. And in that case, the retroviruses came from another part of the genome of the mouse. But there's no reason, in principle, why these retroviruses can't come from the cow, for example. What they did find is things called retrotransposons, which are jumping genes. They ended up in the genome of the mouse. So this is jumping genes from a cow end up in the genome of the gene-edited mouse, right? And so if this goes on to become part of the breeding stock, you're doing a livestock experiment and goat DNA ends up in a cow genome, you could end up spreading viruses and pathogens from one organism to the other. And in any event, you can spread viruses and pathogens if DNA is basically being transferred inadvertently or solutions are being mixed basically in a way that you never anticipated in the breeding program. So this is a huge wake-up call. And it's, to me it's interesting that when we published the article about the antibiotic resistance genes ending up in the cows, that was quite widely covered in the media, right? We were the first to report it, but then now the BBC has had it, El País, Le Monde, various newspapers and websites in the US have covered the story. But when we covered the story of the bovine DNA and the goat DNA and the random E. coli DNA ending up in the genome of these gene-edited mice, no one covered that story. And I think it's because this is, it totally collapses the argument of the industry that they don't want to be regulated. Basically, they can be left to regulate themselves is their argument. And that argument is now smashed to smithereens as far as I'm concerned.
Well, you're very right to pick up on the lack of interest, shall we say, in this story in the mainstream corporate media. In fact, the latest gene editing story that I can find via statnews.com, new CRISPR tool has the potential to correct almost all disease-causing DNA glitches, scientists report, which says that a new form of the genome editing tool CRISPR-Cas9 appears to significantly expand the range of diseases that could be treated with the technology by enabling scientists to precisely change any of DNA's 4 letters into any other and insert or delete any stretch of DNA, etc., etc. Not a single mention of anything regarding the potential risks of this type of technology, although just for the record, CRISPR-Cas9 is not specifically what you're referring to with this recombinetics problem?
No, in the case of the recombinetics experiment, they used another kind of editing methodology. I mean, basically, it's just substituting one enzyme for another enzyme. There's not a huge difference there. In the case of the mouse genome, where the bovine DNA and the goat DNA and the E. coli DNA ended up in the genome of the mice, that was CRISPR gene editing, and CRISPR is kind of considered to be the methodology that will be most used in the future. But of course, that expectation is totally dependent on which companies live or die and patents and all kinds of complications that are outside of most people's control. So we still, we don't know the answer to that question yet.
Well, one of the factors that you have mentioned here is, of course, the regulatory environment that's going to, in some sense, choose the winners and losers in this burgeoning industry. And of course, as you say, it was the FDA that seems to have discovered this problem in recombinetics gene editing. So tell us about the FDA's role in this, where it goes from here. Is this, I'm assuming they're calling for a stop to all of this? No, probably not. But no. How does this proceed from here?
They're not calling for a stop. In the FDA's paper in the quotes, they're quite encouraging about recombinetics. But what's, one of the things that's interesting is not only are there jurisdictional issues, of different countries, perhaps perceiving that they have different interests in all this issue, fighting in a sense over the future of regulation. Boris Johnson, for example, the new British Prime Minister, he's made a whole ton of speeches about biotechnology and GMOs and how wonderful they are, which presumably are linked in his mind to Brexit, right? Because he's thinking that if they want trade deals no longer with Europe but more with the US, then they're going to have to comply with US systems of regulation. And that means that basically the population will end up eating GMOs, which at the moment they mostly don't do. So there are all these complications, and one of them is that FDA would like to regulate gene editing. The twist to all this is the industry itself would like, for the most part, to be regulated by USDA. So you've got two kind of communities, if you like, who are doing gene editing. There's the communities who are doing mammals, who are doing medical research, who are doing, even human gene editing. Basically, there's a medical community. And then there's the agribusiness community, which is also doing gene editing, whose friends are all in USDA, right? So you've got two communities who would, who some of whom would like to be regulated by FDA, and some of whom would like to be regulated by USDA. And you can interpret this FDA, because FDA didn't need to say a thing, right? They could have just done it and had a private word with these people, but they put a paper online, and which they haven't published yet, very interestingly, but they put it online. And essentially, you can see this as their bid to regulate. Look, USDA is offering hands-off. We're offering hands-on. If you want to go with hands-off, that's fine. But look, we've already shown that hands-on regulation is actually needed. And FDA is proposing a regime of regulation, which is actually, a little bit stringent, whereas USDA is basically going to wave everything through.
And once again, it comes down to bureaucratic wrangling that it seems the general public is several steps removed from. But I do note that you end your article by saying it's not just regulators who need to step up, however. Investors, insurers, journalists, everyone, in fact, should be asking far more questions of the scientists and companies active in gene editing. Otherwise, boom is likely to stray into bane. So on that note, what is it that people in the audience can or should be doing to involve themselves in this issue?
Well, it's not so easy to get involved. I mean, there are sometimes public comment periods which agencies open. I wish I was better informed on whether there was any public comment situations coming up or ongoing at the moment. I tend not to contribute to these things because I'm kind of at a disappointed. I don't see much hope in these agencies really. But the public, first of all, it would be great if the public would share information about skepticism, on social media or emailing journalist friends or just sharing with friends. All this kind of background information is really important, that the population has some level of understanding because, in future, the biotech industry imagines that all your food and all your medical products and all kinds of weird stuff that will show up in the stores, like microbes in your yogurt and so forth, and stuff in your beer, will all be gene edited. And the more people know about gene editing, the better, basically, as far as I'm concerned, because basically an informed audience can protect itself, and an uninformed audience has no chance, really, of protecting itself. So there's things like that.
It's a good point that public opinion has a large role to play in this because, as we've just seen with the Monsanto trials and some of the information that's come out from them, Monsanto did have in place a coordinated effort to sway online opinion by actively participating in comment forums and other such things to a crisis response team. Anytime Monsanto was mentioned on the internet, it was like conjuring a demon. They would come up to try to comment on those articles and to sway public opinion. They wouldn't do so if public opinion wasn't important in these issues.
Yeah, that's precisely right. I mean, one of the reasons why public opinion, this is one of the reasons I work in the food space, is because, there's some chance that the public can basically vote with its dollars. In many spheres of public life, if you want the different amounts of money to go to the Pentagon, or different amounts of foreign policy to operate differently. There's the avenues that people have to intervene in that process are limited. In the food system, people are always sharing information at the dinner table. They're sharing information with their friends. They can choose to buy non-GMO food. They can choose to buy organic food. These food differences, provide an opportunity for people to basically vote with their dollars. And I'm the last person to say that, government shouldn't be doing this, but there is a role for the public in the food system that there isn't in many other political domains.
All right. Well, one way for people to actually inform themselves on these issues is, of course, to follow Independent Science News. Tell us a little bit about your organization and what it does.
I used to be a genetic engineer, and together with Alison Wilson, we formed a nonprofit, and eventually this led us to publish the website Independent Science News. And what we do there is basically cover issues to do with science in the food system. So we talk about pesticides, we talk about regulation, we talk about GMOs, we talk about sewage sludge, and we have resource pages and really, really good quality information, so that people don't have to be misinformed about and can find a place where there's good scientists writing about sensible stuff, and we get invited commentaries, and we try to get the best people to write about these really, really important issues.
All right. Well, I think we're going to leave today's conversation there, but of course the links to all of these specific articles and to your website in general will be in the show notes at corbettreport.com for those who wish to follow them. And Dr. Jonathan Latham, I hope it will not be 4 years until our next conversation, so please do keep us informed of the latest developments.
Well, thanks for having me on, James.
The Corbett Report is brought to you by you. Your support makes the Corbett Report possible. Sign up for the subscriber newsletter or purchase a DVD at corbettreport.com/support.